Welcome to Ekam IAS Academy. Today, the point of discussion is an interesting technology that helps in designing tailored medicines. Yes, you have heard it right. In future, we are going to have tailored medicines that are individual specific. That means they'll suit your genetic makeup, they'll suit your bodily requirements and symptoms. Yes, it is possible in future through a significant technology that is biotechnology. And today the point of discussion is about genome sequencing, which is a significant milestone in the process of biotechnology. So without any further delay, let's head to the point of discussion. Here you can see what are the points of discussion of this particular discussion. It is about 10,000 genome project, the main title of the discussion, and why it is a news we are going to look into, and what is the detail about the project, and what is meaning of the genome, and we'll understand the terminology of genome sequencing also, what is the process involved in it, how it will work, and what are the applications of genome sequencing along with the significance or importance of such genome sequencing. Why do we need to sequence our genomes? So then we'll see the conclusion along with some practice question for prelims as well as mains. So here, the syllabus mapping, as you know, it is finding its relation in science and technology, that is in GS paper 3 of your mains under biotechnology specific chapter. Then, if you see the context, so why we are discussing about this 10,000 genome project, that is, here you can see the Department of Biotechnology. Do remember, this is an autonomous body, or you can say autonomous department under Ministry of Science and Technology. So in the recent past, it has announced the successful completion of 10,000 genome project. So that is what we are going to discuss. So we are going to discuss what is this project entail, and what are the main objectives, what are the targets, and what are they going to do as part of this project. Before we proceed further, we just need to understand what is this Department of Biotechnology doing in India. So if you have to remember about DPT, you just remember it is under Ministry of Science and Technology, and also it is very important in the recent past because DBT is playing a major role in different things. That is very important for your prelims perspective. Just like a revision, I'm just highlighting the major achievements of DBT in the recent past. So first thing is India's indigenous HPV vaccine. If you have remembered, human papilloma virus vaccine for cervical cancer is also developed by Department of Biotechnology. So there is an important role of DBT in this particular domain. Then you also have to remember to the COVID times that world's first and India's indigenously developed DNA vaccine, that is Zycovid, if you have remembered, that was also under this DBT role only. Then you have India's first intranasal, through nose we have administered COVID vaccine. So what is the name of the vaccine? Answer in the comment section. That was also administered by this DBT. At the same time, booster vaccine during COVID-19. Not only these vaccine related developments, but also in genetically modified engineered crops also, there is an important role that DBT plays. At the same time, it is also conducting a national biopharma mission. So these are the important things which you need to recollect when you're just hearing about DBT or Department of Biotechnology. So then, as we proceed further, you can see what is this Genome India project. Before that, okay, so this 10,000 genome project is like a sub program, you can say, under a broader program called Genome India project. So first thing you need to know is this is a national project launched in 2020. So in 2020, that means almost 3 years back, we have launched this project. Mainly, it is funded by Department of Biotechnology. It is spearheaded by Indian Institute of Science, that is headed in Bangalore. So, under Indian Institute of Science, there is a Center for Brain Research. You can see here the Center for Brain Research. It is mainly involved in this research. So, what is the main objective here? The main objective is to identify genetic variations. It means, every person genome, okay, almost, human genome is 99% same, we say most of the times, but that 1% difference is making our physical characteristics, mental characteristics, and all these things as different, right? So how that 1% gene or gene genetic makeup is varying, all these things will be studied under this project. Okay, so we'll understand more in detail further. Then this project is mainly conducted in two phases. First phase, they are going to identify genetic variations in 10,000 individuals across India. That means different parts of India they wanted to identify. In the second phase, they wanted to collect the samples from patients who are suffering from cardiovascular diseases, cancers, mental illness, and all. So they'll understand which gene is being responsible for causing these diseases and all. That is being proposed in the second phase of the project. Okay. So now we are limiting it to the first phase because we successfully completed the genome sequencing of 10,000 individuals across India. Then what is the main objective or aim of the project is, here we wanted to create a like you can say, we wanted to construct a database, different database you can say, of whole genome sequences. That means almost all the major Indian genes we wanted to sequence, and we need to understand how their genetic makeup is. Okay, so what is the main characteristics of their gene, and all these details we wanted to understand. But at the same time, we also wanted to develop predictive diagnostic indicators. That means with the help of genetic makeup, we can better diagnose in the future the diseases which they are susceptible to, and all these things. Okay, we can design medicines in a way that will suit their gene or genetic makeup. In that way, we can plan. So, for these objectives, this particular project was launched. So, you can see the main applications of the project are finding in agriculture, biotechnology, and healthcare. So, in these three sectors, we find the applications of this Genome India project. That means if we have conducted successful genome sequencing in human related projects now, then we can even extend that to different bacteria, fungi, and virus also. And in healthcare also, we can use this particular technology and particular project details, and we can enhance our healthcare also. That is why the main object of this project. Not only this, we need to understand till now how many countries have successfully genomed their sequences means, sequencing of genomes means, till now only three countries have achieved this feat. That is USA, UK, and China. Only these three countries have successfully sequenced their 1 lakh population genome. That means here we have completed only a smaller domain, you can say, that is 10,000 sample only. But these three countries have sequenced almost 1 lakh sample. Okay, that means among 1 lakh population in their countries, they have sequenced their genomes. That is one important thing you need to know from examination perspective. Then we'll understand it better. Then you can see here what is this 10,000 genome project. Okay, this is project Genome India project is the broader project, out of which a subcomponent you can say, that is 10,000 genome project, which is being conducted in phase 1. So this is an initiative of Department of Biotechnology, as I told you, mainly funded by Department of Biotechnology. Aiming is to sequence almost 10,000 Indian genomes. So they wanted to sequence 10,000 genomes. The objective is to identify variations in the genes so that we can predict the diseases, and we can also develop the medicines so that it will suit individually. So that is what we discussed in the beginning, with tailored medicines. That is the main objective of this project. Then we need to understand what is a genome. So for that, you need to know basic components of a cell and all. So cell means what? Cell is a basic functional unit of life or any living organism, to be precise. And these are the basic building blocks. So every human organism, cell is made up of tissue. Again, tissues are made up of cells. Again, with the tissues, organs will be developed. Organs will form into organisms, like that. Again, it will extend, right? So fundamental unit of any human body or any living organism, we call it as a cell. Inside a cell, we have a nucleus. You can see in the image also here, there is nucleus. Inside nucleus, we have chromosomes. So in human body there are 23 pairs of chromosomes. That means totally 46 chromosomes are there. Inside every chromosome, again, there will be DNA. What is DNA? Deoxyribonucleic acid. What is DNA? DNA is nothing but deoxyribonucleic acid. So it is a double stranded molecule, and this is forming the genetic makeup of any living organism. So we'll understand it better. You can understand, right? In a cell, there will be nucleus. Inside a nucleus, we have chromosome. This chromosome will be normally present in mitochondria. Mitochondria is considered as powerhouse of the cell, right? So inside this chromosome, there are DNA. Inside DNA, again, there will be genes. So that is forming this individual differences among the living organisms. That is mainly responsible, right? Then we'll understand human genome. So for this to understand, you need to know what is the meaning of the word genome. Genome is nothing but a complete set of DNA instructions. So if DNA instructions present in a cell, so based on these instructions only, the human body or the human individual will develop and it will function accordingly. So it is nothing but it is a composition of small chromosomes which we have seen. These chromosomes are present in the mitochondria, and mitochondria is in the nucleus of a cell. So in this way, genome is nothing but a set of complete instructions that are present in the DNA of a cell. Okay, this is what you need to know. Then second thing you need to know is these are made up of chromosomes. Okay, so then you need to understand what is the function of human genome. Human genome contains all the information that is required for the person to develop and to function. That means how human beings will develop over a period of time, right? Physically and mentally. So this development is also mainly due to this human genome itself, right? Then we'll understand what is there in the DNA. So coming to this, DNA carries the genetic information, right, that we inherit from our parents, right? So DNA consists of double-stranded molecule. As you have seen in the image here, double-stranded means this one. Okay, so you can see here, double-stranded molecule which is built of four bases. Four bases are adenine, cytosine you have, guanine and thymine. Okay, so four nucleic bases you can say, nucleoid bases you can say. But all the time you have to remember is this A and T will be clubbed together and they will be together, and C will be with G. Okay, so these are fixed pairs you can say. That means adenine and thymine will be together and cytosine and guanine will be together. So this is what genetical makeup will be there most of the times. Okay, that means all these, how many will be there means again almost 3 billion bases will be there in a genome. So that is what you need to know, an interesting fact. Okay, then we'll understand what is the meaning of the word genome sequencing. So for this to understand, all organisms like bacteria or plants, or you can say vegetables, I mean they're plants, and even mammals also. Mammals means which will give birth and they'll feed their younger ones, you can say. So they have unique genetic code. They will be having genetic DNA that is very unique to them, right? So they will be composed of these nucleotide bases like ATCG we have seen now. So this basic DNA will be comprising of these nucleotide bases ATCNG. So they will be very unique DNA. So this uniqueness, how they will be uniqueness means this uniqueness is attributed to the sequence of bases. That means we have seen A will be with T, C and G together, right? So here you need to understand this uniqueness of DNA will be attributed mainly to the sequence of bases. For this to understand, you have to see the next image. So here you can see in this image, you can see in the third step CNG, CC, AT, like this, there is a sequence. So in which sequence they are placed or in which sequence they are observed, based on that there will be uniqueness in the DNA. So this is what we consider as DNA sequencing. That means in DNA sequencing or genome sequencing, you will determine the order in which these nucleotide bases are present in a genetic makeup of that human, okay, or the organism. So sequencing is nothing but we'll decode this order of the nucleotide bases which is present in the gene of that particular organism. So this is a laboratory procedure. So how this will function now we'll understand. So for this to understand, you need to know the process, right? So first, how it will work means the first step you can see cutting the DNA. So how you can cut this DNA means the first step or the first process we call this as DNA shearing. Shearing means here molecular scissors will be used to cut the DNA. We have seen DNA means like this, we have double-stranded molecule is there, no? So this will be cut. That means which part we wanted to sequence, that part will be cut using a molecular scissor. So if you have remembered in the recent past, we have a CRISPR-Cas9 technology in which that Cas9 is a protein or a molecular scissor which will be used to cut the gene, right? Gene editing technology we have seen. Same type of molecular scissors will be used here to cut the DNA. So after cutting the DNA, that is what we refer to as DNA shearing. Then second step is called DNA barcoding. In this process, what happens? Normally barcoding means when you visit a supermarket and all, normally when you're billing, the person will barcode or scan the barcode. So with the barcode, you'll identify which is the product and how much price is that product listed. So these things will understand, right? In the same way, DNA barcoding is nothing but a method of identification of species using a short DNA segment. So now we have cut down the DNA. That particular DNA segment will be taken and they will be identifying that particular DNA segment and we'll understand from which specific gene it was taken. Okay, that is in the second step. Then third step is nothing but DNA sequencing. You can see here, this is the DNA sequencing method. In this process, what happens is the barcoded DNA will be there. That barcoded DNA from multiple bacteria or from multiple organisms will be combined in a sequencer, or you can say machine will be there. In that, they'll combine into a single unit. Then all these sequences will be placed together in a computer software. Based on that, we'll understand what is the genome sequencing, or the same thing will be used in genome fingerprinting technology or DNA fingerprinting technology also. The fourth step, after the DNA sequencing, the fourth step is nothing but data analysis. You can see the final step here. You can see that is nothing but to compare the sequences from multiple organisms and identify how they are different. Okay. So if you just say like we'll understand, and most of the people will say like chimpanzee and humans, they have similar type of genetic makeup. So how much similar we have and what makes us different from the chimpanzee genome and all. And individual, every individual, although we inherit some characteristics from our parents also, we have our unique you can say unique physical characteristics, right? So how we are having this uniqueness? So what is the system of arrangement of these nucleotide bases in our genetic makeup? All these things will be understood by this genome sequencing technology. So now we have understood how this process is working. Okay, that means we have seen four steps involved. First one is cutting the DNA, that is called DNA shearing. Second step is barcoding the DNA, so we'll understand from which gene we have taken, that is in the second step. Third step is DNA sequencing, so we'll arrange these into one sequence, and then we'll understand by data analysis, that is using a computer software. So you can understand from this image. So first thing, as I told you, this is the first step. Okay, cutting down DNA sharing. So this is the second step, DNA barcoding. Then this is the DNA sequencing you are using, and data analysis in the fourth step. So this is the entire process how the genome sequencing will be done. Okay, right? Then we'll understand what are the applications of this particular genome sequencing. So how it helps, you can understand here, right? So you can see it helps in preconception and prenatal testing. That means even before the birth of the baby girl or baby boy, so we'll understand how their health is there and what is their genetic makeup. Okay, is there, if we identify any defective gene, then we can use this CRISPR-Cas9 type of technology, and we can cut that particular gene, and we can better the health of the particular baby. So that is one main application of this genome sequencing. Then newborn screening, that is also helping understanding and determining the characteristics and the genes of the newborn. At the same time, we'll also understand the disease susceptibility. That means how much vulnerable of one particular gene, okay, so will be with respect to particular diseases, how much vulnerable they are. All those things can be understood. Then last application, you can say screening and diagnosis. That screening and diagnosis means with the genome sequencing, we can identify this basic technology. With this basic technology, we can understand how the screening can be done of diseases and how diagnosis has to be done. Here you have seen two terms, prognosis and diagnosis. For this to understand, you need to know what is the difference between prognosis and diagnosis. Prognosis is nothing but determining how the disease will progress in future. That means if you're attacked with flu, some symptoms will be developed, and there will be some progress in the disease. Later on, it will subside, right? Same way, what it will do on your body or these things, that is the prognosis. Diagnosis is determining which disease you have. So you will be identifying whether it is a flu or whether it is variant of flu. These type of things will be part of diagnosis. Prognosis means how it will proceed further, okay? How the disease will progress in the future in the human body. That will be determined under prognosis, and this is diagnosis. Okay, so these two things you need to understand. Then monitoring the disease burden and recurrence. So what is the disease burden of a few genetic makeup, and some people, okay, so generally there are some people who are not you can say not vulnerable, or who are not acceptable to anesthesia and all. Okay, so if they are given anesthesia, they'll have some side effects and all. So that also will be understood. So which gene is mainly responsible for these things, we'll understand better with this genome sequencing. So these are all the applications of genome sequencing. Then what is the significance or importance of this project is, it is helping the researchers in identifying the genetic variants. That means what are different genes we have, and which gene is being responsible for what type of features and all, we'll understand. And uncovering the disease history, so we can also understand the disease history of which gene is causing which particular disease and all. So these type of things we'll understand. It also helps in treatment of diseases, you can see here. and like we can understand, right? And in case of this particular gene is causing this disease, we can better understand that, we can cure that particular gene, and then we can lower down the disease also. In that way, disease burden also can be lowered, and then you can see helping in customizing the drugs and therapies. That means, as I told you in the beginning only, tailored made medicines, right? Individual specific medicines can be given in case if we understand the genetic makeup of the individual. So this is all the importance of the project. So then what is the way ahead? How this helps is, when you are understanding the genetic diversity, it always helps us in understanding great information, right? So about the human species. So we can go to the micro level and we can better understand what is causing this diversity, and if any problem arises because of this genetic variation, we can solve it before the hand. Okay, that is what the way forward or way conclusion you can say for this particular topic. Then, as we have come to the end of the video, let's see the prelims question for practice. So here you can see, consider the following statements. DNA barcoding can be a tool to assess the age of a plant or animal. Then distinguish the species that look alike. Third, identify desirable animal or plant materials in processed foods. Which of the statements given above are correct? You have one only, two only, three only, one and two, and two and three. So this is the practice question for prelims. Then practice question for mains. You can see, genome sequencing has revolutionized biological research and healthcare, providing unprecedented insights into the structure and function of genomes. Discuss. So this is a mains question for practice. As we have come to the end of the video, today we have discussed what is this Genome India project, what is the part of this project, that is 10,000 genome project. We have seen what is actual genetic makeup of an individual, right from the cell. We have seen what is genome, then we understood what is genome sequencing and how this process works. We have seen 4 steps that are involved in the process, and also we have seen the applications of this particular technology, and also we have seen the significance of this project. So this is all that we have discussed as part of this discussion on genome sequencing. Thank you for watching. I hope you found this video useful. All the very best. Have a good day.